Hello and good evening, everyone. And I extend a hearty welcome on behalf of SigTuple to an amazing seminar session today of what's behind the dots. So let's strip the jargon and get into the workflow of flow cytometry, the instrumentation and optics. We all know that flow cytometry is a very powerful tool in hematology, and over the past few decades, it's actually transformed to become a cornerstone in hematology, oncology, and clinical diagnostics. And in today's session, we have none other than our esteemed speaker, Dr. Thulasi Raman Ramalingam. He is the consultant at the Central Reference Laboratory at Apollo Chennai, and his expertise extends into MRD, immunophenotyping, and rare immunodeficiency diagnostics. He has over 30 peer-reviewed publications and multiple scientific awards for scientific innovation in flow cytometry. He's also a faculty member of national level cytometry conferences, and he has been featured on the cover of International Clinical Cytometry Journal as well. His work has been featured out over there. He's played a pivotal role in developing and reporting advanced diagnostic assays for leukemias, lymphomas, and plasma cell disorders and primary immunodeficiencies. What makes him amazing is that he has a brilliant academic mindset and diagnostic acumen, which has been shaped over a long period of time by both clinical practice and research. He continues to be a prominent figure in clinical flow cytometry in India. Please join me in extending a warm welcome to Dr. Thulasi Raman Ramalingam. We welcome you, Dr. Thulasi, and over to you.
Okay, thank you so much, Dr. Renu, for giving this opportunity. Really, it's a pleasure. And I want to thank you and your SigTuple team for giving this opportunity. And I also really want to thank the participants more. You know what? Because Saturday evening, if they're coming here to learn flow, really, a big salute to you guys. Because Saturday evening, if the same link is shared to me, it's doubtful that whether I will log in. Great.
Absolutely.
But what I can promise you is that for the next 1 hour, you will enjoy. So what we are going to do is to peep our head into a flow cytometer and see how it's working. So I want this session to be more interactive. Okay. So we are going to see how a flow cytometer works. So that's what I'm going to exactly trying to do. I want you all to get into the flow cytometer, see what's happening inside. Right. So normally I'll just start with a story, where I have a son. Whenever I go out, children are normally demanding, especially boys. You take them somewhere, they say, put a condition. If you get me, I'll be silent. If you're not going to get me this toy, then, I'm going to create. So normally we'll get him a toy. And also some days back, I got him this helicopter, right, which is lightning and it flies. So for the first day, what he does is he just keeps it safe. He doesn't allow anyone to touch, even me, who bought him that. Second day, he used to admire it, see how it's flying, everything. And third day, what he does, he slowly dismantles it. You know why? He is curious to know what's inside, how it's flying. Okay, so that's the same curiosity what we all have. So how come flow can generate so beautiful data from which clinically we give great diagnosis and everything, but how does the machine does it? No, so fascinating, right? So that's what I'm going to do. Before I go, just to give a takeoff. So why you really need a flow cytometer? Anyone? Can the participants unmute and answer? Do we have that option?
Doc, I think we have that option, right?
Yes, ma'am. Sure. Yes.
So Madam, we do have, but we want sir to speak more and we want to be only a listener.
Harivarsan, yeah, we'll do that, but you can just say, you want to really have a flow cytometer. See, because microscope is gold standard. Still, microscope is gold standard. You can study cells with microscope. But why do you really need a flow cytometer? Right? The problem with microscope is that, of course, I understand it's gold standard, but you cannot count the cells with microscope. Right, and it is subjective. What appears like a reactive lymphocytes may appear like a blast to someone, and you cannot analyze large number of cells. As I say commonly, how many of you have encountered a stem cell when you are seeing a peripheral blood smear? It is not like it's not there; it's there, but you have not seen that many leukocytes to have a chance of encountering that stem cell, because that stays in, say, 1 in 10,000 cells. We never go up to that level of seeing the cells, right? And so that's the reason why we cannot study rare cells with microscopy. And beyond all that, it's time consuming. So whenever I give a hypocellular marrow or a leukopenic blood smear, I normally see my residents don't type 100 cells differential. They just type 25 differential and into 4 they do, right? These all happens because it takes time. But these all can be bet or overcome when we use flow cytometer, which is a very, very rapid tool. And it can analyze cells at a speed of, say, 10,000 to 1 lakh in 1 second. So it is that fast. That's the beauty of flow. So how does flow work? Very simple. This is what the image I show them, very clearly understood. So as the name says, flow cytometer. So you are measuring or studying the cell when it is in a flowing stream across a light source. Correct? Very simple. So you are studying the cells in a flowing stream across a light source. So when light hits the cell or the particle, what happens? Basic. It scatters. Correct? It scatters. So with that scatter, we can able to study the physical properties. So that's what happens. So let's assume that this is the cell which is flowing. Okay. I'll come back later with what is there in this and what is all flowing. But cell is flowing through a liquid stream, and you have the laser beam or the light hitting the cell, and two things happen. Like the scatter happens. The scatter is again of two types. One is the scatter that happens in the same direction of the light source. What we call it as the forward scatter, right? And the scatter of light which happens perpendicular to the light source, which is called side scatter. So what does the forward scatter imply and what does the side scatter imply? Forward scatter will tell you about the size of the cell. Okay, just assume this one. If it's a smaller cell, you get a smaller forward scatter. If it's a larger cell, you get a larger side scatter. As simple as that, right? It's a common sense, right? If you have a small particle going across the laser beam, the light scatter in the direction of the light source will be smaller. If it's a large football size going across the laser beam, the scatter of light across the same direction of the light will be huge. Correct? And similarly, so forward scatter is basically proportional to the size of the cell, right? And how about side scatter? Now, what does side scatter tell you? So I told you that side scatter is the light that is getting scattered in a perpendicular fashion across the light source. So the light source is from this direction, it is perpendicular. So the scatter which is collected perpendicular to the direction of the light, incident light, is called side scatter. So what does your side scatter say? Forward scatter size. What does the side scatter say? It says about the internal complexity. So for example, if you have a promyelocyte, now the side scatter will be very high because it has so much of azurophilic granules in it. Correct? Whereas if a lymphocyte is hit by the laser beam, the amount of side scatter will be low because there is minimal cytoplasm and there is no much of granules or vacuoles or nothing is there, right? So side scatter tells about the internal complexity of the cell, right? I repeat, forward scatter will tell you about the size and side scatter will tell you about the internal complexity, the granularity, vacuolations, and so on, so inclusions, etc. Now this is what, so this is how a pulse is generated. So this is the laser beam and the cell goes through, you get a pulse. Okay, for every cell there will be a pulse which will be generated, and finally a data will be given out, right? So I'll be going in detail later, but now, so with just forward scatter and side scatter, now you can able to make out the cells or populations, right? So for example, and remember, every dot is given by a cell. I repeat, every dot in a flow cytometry dot plot is a data given by a cell. Remember that. And this dot, we say, don't say dot, we in flow cytometry language, we call this as events, right? And events with same characteristics will tightly fall, right, close to each other. And that we call it as population, right? Or a cluster. So here we have a cluster, here we have a population or a cluster, and here we have a population or cluster. Now we have forward scatter on the x-axis and side scatter on the y-axis. So what is this population? This is a population which has very low forward scatter and very low side scatter. So in a peripheral blood setting, this is lymphocytes. Now how about this one? This is a population which has relatively higher side scatter than lymphocytes and relatively higher side scatter than that of lymphocytes, and these are monocytes, right? These are monocytes. And this one, has higher side scatter than that of monocytes, but forward scatter almost similar of monocytes, and this is neutrophils, right? So that's how you call the population. So I repeat these terms, maybe where cytometrists use these terms, population, clusters, events, it all means this is the actual points behind that. Okay, so this is about thing. Now one question to you, like, so you have a lymphocyte cluster here. So can you able to further say whether it's a B lymphocyte, T lymphocyte? Can you able to say that with forward scatter and side scatter, which is basically your physical characteristics? Can you able to say whether this lymphocyte is a B lymphocyte or a T lymphocyte? Can you able to say?
No.
We encourage the participants to answer the question.
Yeah, you can just unlock and you can answer. Come on, let's be interactive.
Yeah, it doesn't matter if it is not right or wrong. It doesn't matter at all. Dr. Thulasi is here to teach all of us.
Yeah.
So,
So we are here to learn, right?
Go
Even
ahead.
I'm learning every day.
Yeah, that's right. Please go ahead.
Yeah.
Yes.
So
Yes, sir. We can differentiate the B lymphocytes and the T lymphocytes because of this position of the scatter.
Position of the scatter, but no, ma'am, you can't, because do you physically see the T lymphocytes and B lymphocytes are same? Morphologically, it will be same. If you see a slide of if you say a peripheral blood smear and you find a lymphocyte, can you able to say it's a B lymphocyte or T lymphocyte? You can't say, because B lymphocytes and T lymphocytes by physical properties or by appearance is the same. So how do you differentiate? So now, apart from the physical properties, you need something extra to differentiate them. What are those? That is, you need to explore the antigenic properties. Now, how do you want to explore this antigenic property? Let's assume, so this is something done with fluorescence in flow cytometer. How it's done? Now let's assume this is a B lymphocyte, and let's assume this is a B antigen, like CD20. Okay, CD20, and this is, we use an anti-CD20 antibody tagged with a fluorochrome. Okay, fluorochrome is a tracer element. Okay, so when you add, what happens? This anti-CD20 antibody tagged with fluorochrome will bind only to the B cells and not to T cells. Agreed? Right. Okay. So now this cell, if it goes across the light source, that is the laser, what happens now? This fluorochrome gets excited and it will give signal in a different color, okay, which will be captured by a detector, which is present in the flow cytometer. So that's how a flow cytometer, apart from capturing data from the physical properties, that is forward scatter, side scatter, it also captures data about the antigens and proteins present in the cell to diversify the populations, which are similar by physical appearance. Beauty, right? So certain things we find difficult to understand, but you think about the person who discovered it. No, hats off. So that's how the flow works. And the beauty of flow is that, in contrast to your other immunophenotypic techniques like IHC, multiple antigens or proteins can be studied simultaneously in every cell. It's a beauty, right? So what I've shown here as an example is just one antigen. Similarly, simultaneously, you can study almost like 13 antigens with a 13 color flow cytometer at a given point of time in one single cell. That's the beauty of flow cytometer, as I've shown here. I have shown here with 5 markers, but similarly, you can study 13 markers, 20 markers based on the configuration of your flow cytometer, right? So what happens in a flow cytometer is that when you put the cells in the flow cytometer, which has been stained with the antibody, as I just shown you, okay, so as it moves across the laser beam, what happens? Now, 2 things happen. One, the cell will be hit by the laser or the light source, and scatter will happen, and the forward scatter and side scatter will be collected. Parallelly, the other thing also happens. What happens? The light hits the fluorochrome, which is attached to the cell, right? And a fluorescent emission will happen, which will be captured by the detector from the same cell. So now you are parallelly capturing the physical properties by scatter, forward scatter and side scatter, and you're also exploring the antigens of proteins present in the cell using an antibody tagged with the fluorochrome, right? So you're capturing both physical properties as well as the antigens and proteins present in the cell. And now you can diversify any population. Let it be a lymphocyte, not only as B lymphocytes and T lymphocytes. You can further characterize T lymphocytes as helper T cells, cytotoxic T cells. Further characterize helper T cells into naive, central memory, effector memory, terminally differentiated, T regulatory cells, and whatnot. And you need this because your immune system is so diverse. You need such a technique to completely study it, right? Is there any doubts till now? Can I go into the flow cytometer now? Because I want to give this takeoff. You need to understand what is the principle before I go into the instrumentation. Any doubts till now?
You can
I'll
also
be
type
happy
in
to take your
questions in
questions.
the chat. Yeah, you can
can,
also
okay,
type
fine.
the question in the chat in case you do not want to speak it out.
Exactly.
We will read it out as well.
Yeah, that is also a great idea. If you doesn't want to speak out, like if you're in work or something, you can put parallelly in the chat box. We'll take it up. Right. So now let's go. So this is what is happening. So we have cells which is stained with fluorochromes, that is antibody tagged with a fluorochrome. Okay, and as it goes in a single file pattern, I'll tell you how this is achieved. The light source, commonly a laser, hits that, and what happens? Remember, two things happen: scatter, right, which will tell you about the physical properties, the forward scatter and the side scatter. Parallelly, the fluorochrome, which is attached to the cell via the antibody, will give you signal, fluorescence emission, right? And that will be captured by the detectors. So you're exploring the physical property of the cell as well as the antigens and proteins present in the cell parallelly. And that's how this flow cytometer is quite different from your cell counter, which you're using in your common laboratory. So I commonly call this hematology analyzers, the automated hematology analyzers, 5-part, 7-part, as a common man's flow cytometer, because they use scatter properties, but they don't use fluorescent properties. So the modern flow cytometer is one which combines both the scatter properties as well as the fluorescent properties, or the antigens or proteins present in the cell. Right? So 2 parameters are recorded from each cell. Remember, each cell. So now let's go into the components of a flow cytometer. Right? So this is a common simple layout telling about the components of the flow cytometer. So first you have the flow chamber, which is the heart of the flow cytometer. Commonly people call it as flow chamber or flow cell. I'm not going to use the term flow cell because you may confuse it with biological cells. So I'll use the term flow chamber. So this is the heart of the flow cytometer where the cells are injected in, right? And you have something called the sheath fluid, which will be flowing into the flow cell. I'll give you this in more detail. And then you have the light source, which is very important. The light source, which is used to hit the cell, right? Or is the light source, okay? So then you have optics. The optics are basically filters. Okay, I'll tell you what's that. Then the filtered light should be captured, and you have use detectors for it. And then electronics, where the light which is captured by the detectors will be converted to electrons, photons to electrons, which is done with electronics. And then you have the software to analyze. So let's start with these two, flow chamber and fluidics. Okay. So what is this fluidics or the flow chamber does? So you need to see this image in a 360 degree, though I have shown it in a 2D way. Imagine as a 3D. Okay. There is a funnel. Okay. And the funnel has a continuous flow of a fluid called sheath fluid, SHEATH, sheath fluid. Okay. The velocity of the sheath fluid will be higher. Okay. When in such high running sheath fluid in a funnel, the cells or the particles will be injected in the center, in the central core, or injected in the center. Now, practically imagine, so in a 360 degree, in a 360 angle, you remember, you just imagine when the sheath fluid is running so fast, okay, what happens in this area? The speed increases or decreases, or the velocity increases or decreases? When the sheath is running so fast in the funnel, in this area, the speed of the sheath fluid will, due to funneling, what happens? It will increase, correct? So when these cells are injected in the center of such a fast moving sheath fluid in a funnel, when it comes to this area, because of the increase in velocity, all these cells which are randomly placed will come in a single file pattern, that is one after the other. This is called Bernoulli's theorem, right? So there is a outer faster moving sheath fluid, and there is an inner core of cells or particles. When they come to this area of funneling in the flow chamber, due to Bernoulli's effect, the cells which are random becomes in a single file pattern. This is called hydrodynamic focusing. This is the first tick for a quality check in your flow cytometer. If hydrodynamic focusing is not done properly, cells will not go in a single file pattern, and obviously the data what you collect is all useless. Okay, so people call this as stream within stream, because the central stream is by the blood sample or your cells or particles, and the outer stream is given by the sheath fluid. So which moves faster? The sheath fluid moves faster than the blood sample, which contains cells, right? And because of this funneling which happens in the flow chamber, the velocity of the sheath fluid increases, exerting pressure on both sides, making the cells to pass in a single file pattern or one after the other. And this is called hydrodynamic focusing, right? The flow chamber or the flow cell is a place where the cells are delivered in a single file pattern. Because why you want the cells to go in a single file or one after the other? Why can't it go in 2 and 3 as a clump or together? Because the ultimate idea is the laser beam should hit one cell at a time. Then only it will be properly studied. Then only the scatter will be pertaining to that of the cell. Even see, if you ask me to just observe one person, I can observe him properly, say from the head to foot. If you give a group of people and say you observe, you observe everyone, my observation may not be that great, right? So similarly, the laser beam should hit one cell at a time for a proper scatter and proper fluorescence, or capturing the fluorescence and emission, right? So that is a reason behind the importance of hydrodynamic focusing, to make the cells or particles to go in a single file pattern.
Dr. Thulasi, we have a question on the chat box.
Yes.
ESS, is the sheath fluid in contact with the cell line?
Yes, it will be in contact with the cells. So see, why we have shown this partition is because to make things clear. But when it comes to this area, right, you can see there is no, they will be in contact with each other. The sheath and the cells will be in contact. It is not like it will be divided. Then how come the sheath can put the cells in single file, right? It will be in contact when especially in this area, in the funneling area. Okay, right. So the flow cells are designed to accelerate and space out and focus and align particles or cells, and this is achieved by funneling. And you need to remember the flow cell dimension of your instrument. That is important. Why? Now, for example, I want to study a large human oocyte, which may be the biggest cell. Okay, I need to check whether that oocyte can go inside the flow cell. If it is too big enough, it may not able to go inside the flow cell, right? And you may not able to study. So the common conventional flow cytometers flow cell dimension is between 430 micrometer to 180 micrometer, and you can study cells ranging from 5 to 150 micrometer. So most of the biological cells are within this range, and we can study those cells by flow cytometer, right? Because this is the dimension. This is the dimension which I'm telling, 0.5 to 150 micrometer. And this dimension is the total dimension, where including the sheath, the area where the sheath flows, right? So this total dimension is this one, and this dimension is this one, right? So this is what happens. So I'm just showing this animation. So these here cells are random. As they come down, okay, because of this funneling effect, they are made to pass in a single file pattern. See, there is a faster moving sheath, and this is the sample core. And you can see here the cell and the fluid sheath is in contact with each other. And because of this funneling, it makes to make the cell pass in a single file pattern, which is called hydrodynamic focusing. Now, what is this sheath fluid? What is it really? Is it something, a magic liquid or something? Definitely not. It is nothing. It is just a water, or it can be a buffer. Okay, so the sheath is nothing but it's just a water or a buffer. Okay, it should not be a hypertonic water. Remember, it could be a distilled water, okay, or a buffer solution, okay, which makes the cell to pass in a single file pattern. Clear? Everybody clear with hydrodynamic focusing? Anybody has a doubt here? You can put it in the chat box or something. So if you're clear with hydrodynamic focusing, then I can go. Okay, so I repeat, a faster moving sheath fluid, okay, makes the cells to go in a single file in the funneling area because of the increase in velocity of the sheath fluid. And this is achieved because you want one cell to be interrogated by the laser beam at a time, right? Right. So this is what happens. So see here we have a See, now you assume two flow cells or flow chambers. This is one flow chamber. This is another flow chamber, A and B. In A, you don't have much space for the sheath fluid. As you see, this is the sample core, the sample area where the sample is injected. You don't have much space for the sheath to run. When if a flow chamber is like that, obviously hydrodynamic focusing will not be achieved. Whereas here you see, you have a flow chamber where you have adequate space for the sheath fluid to pass. And this will be passing in a higher velocity. And here you have the sample core where the sample will be injected. And because of this Bernoulli effect, increase in velocity of the sheath fluid at this area, funneling area, the cells will be aligned to pass one after the other. And this is called hydrodynamic focusing. So single file pattern is mandatory, is very, very important for a good flow cytometry data. So for the pathologist out there, when I say single file, what comes to your mind? Lobular carcinoma, histopathology. From here on, when I say single file, what should come? Hydrodynamic focusing or flow cytometry. Okay, great. So now to just show you the layout, because I showed you that this is the flow chamber, this is where the sheath fluid flows, and this is where the sample will be injected, the sample core. Okay, now the pressure or the speed of a flow cell that is getting dispensed into the flow chamber, or the speed of sample that is getting dispensed into the flow chamber can be adjusted with a regulator. Okay, so for example, now if you increase the sheath fluid pressure with this regulator, as I shown here, the amount of sheath fluid which will be delivered into the flow chamber increases. Okay, similarly, when I increase the pressure of the sample regulator, the amount of sample that will be dispensed into the flow chamber will increase. Right now, out of these two regulator, this one you can have a control on, where you can adjust. For example, if you get a hypocellular sample, now you can increase the sample pressure so that more amount of sample can get dispensed into the flow chamber. Because when the sample is so hypocellular and you want to acquire in a low pressure mode, it may take 2 hours for you to achieve say 1 million events or 2 million events. In that case, you can increase the sample pressure so that more volume of sample can be dispensed, right? Whereas the sheath fluid regulator, you cannot access it. Only the service engineer or the company people have can access it. It's almost like defaultly set. Okay, you can access this. So you can acquire a sample in a low pressure mode, in a medium pressure mode, and a high pressure mode. High pressure mode is used for hypocellular samples, and low pressure mode is used in a hypercellular samples, and medium is what we use for regular samples, right? So this is about the fluidics layout, right? The same here. So this is what I mean. So this is sample pressure, which you can adjust, where the users can adjust it. The sheath tank pressure is normally you cannot change it because it's set default, right? And see, now if you acquire the sample in a low sample pressure mode, you will have beautiful flow of cells one after the other. But in a hypercellular sample, say this is the time, Saturday evening, your technician wants to leave. So what they do, if I'm going in a low pressure mode, it's going to take 1 hour time. So I'm going to go in a high pressure mode in a hypercellular sample. Cells, more amount of samples will be dispensed into the flow chamber. And you get, since the sample is already hypercellular, you cannot achieve single file pattern, though you have a efficient sheath fluid flowing by the sides, right? So hydrodynamic focusing is lost and the data will be a crap. Clear? Now, certain technologies are there used in some instruments, like this is a technique which is used in thermo, where apart from hydrodynamic focusing done with sheath fluid, they also couple up with another technique called acoustic field focusing. Acoustic means sound, right? Acoustic field focusing. So apart from the sheath fluid delivering the single file pattern because of the faster moving sheath fluid by Bernoulli's effect at the funneling area, in addition to that, they are also using sound waves. So from both the on both the sides, the sound waves will be liberated. When I say sides, it means all round 360 degree. Remember, I'm showing a 2D picture, but you should see it as a 3D 360 degree. Okay, so the sound waves will be delivered on both the sides, and even 1 or 2 doublets which is failed to make by hydrodynamic focusing by sheath fluid will now made into single file by this acoustic field, because the sound wave will be emitting from this side and also emitted from this side. That makes the cell to go in a center and in a single file pattern. Clear? So this is again some advancements. Instead of the sheath tank where you adjust the pressure, now there is a peristaltic syringe-like model where the sheath and the sample can be dispensed into the flow cell. These are all small modifications, nothing beyond, but the principle is the same, right? So coming to the close of fluidics, in a flow chamber with proper fluidics, what should be achieved? Hydrodynamic focusing, where we achieve a single file pattern. So this is what happens. So you have a faster moving sheath, and in a sample, because of the increased velocity at the funneling area, the cells will move in a single file pattern and they will be hit by a laser beam. Okay, one at a time. Is that clear? Anybody who have doubts with fluidics? Can we go for the light source now? The second component. No doubts? Is there any questions in the chat box?
I don't see any questions in the chat box.
Perfect. So people
Sir,
are
I have one
Yes, sir.
question.
Yes, sir.
So you
Sharing,
told the
yes, sir.
cells. Yeah.
Ah.
Thanks, sir. So
Yes.
I have doubt. So you told that the sheet is not connected with cells, no?
Yeah, no, I said like sheath is connected with the cells.
Oh, sorry. I have connected with cells. But
Yeah, see,
I have doubt if you are using distilled water, no? If if you connect with cells, it will disturb anything. It will lose any.
yeah,
It will burst
so
or
that is true. That is true.
anything.
That is why we normally use a PBS,
PBS,
buffered
okay.
saline, so that the cells are not damaged much. We have experienced like when you use hypotonic water or something, no, the cells tend to rupture out. But when you have
Okay.
a water which is of an equal pH and the same concentration of your cells, obviously the cells can be. So the sheath is nothing, as you know, the sheath is nothing. It is just a fluid which has to carry the cells, make them go in a single file, as simple as that. So that's not like a magic fluid. It's it could be a buffered water, buffered saline. It could be a buffered saline, which we commonly use as phosphate buffered saline, PBS, or it could be some distilled water also. But it should not be too hypertonic for the cells to burst out.
Sure, sir. Thank you, sir. Thank you.
Yeah. So now coming to the next part, very, very important part, light source. Now the first question what I want to ask, which light source you want to really use? Can you use the tube lights what we are using? Or can you use the halogen lamps? Or can you use any other lamps? You want to specifically use lasers. Why laser? Anyone want to try? Why you want to use laser specifically? Why not regular LEDs or halogen lamps or any other light source? Why specifically laser? Because laser has some beautiful properties. Everyone has played with the laser, right? I feel everyone during our childhood would have tried using lasers for multiple reasons. I'm not going into that. But the laser is a monochromatic light. It emits light of single wavelength. And it is coherent. What is coherent? Coherent, these are all physics terms. It goes in a single direction. Third, it is high intensity. It is an high intensity light. So it can travel a longer distance, right? It is coherent and uniform. And it's a thin beam of monochromatic light. So monochromatic means light of one wavelength. And why it should be thin? Because the point where it's going to interrogate is a cell. And the cell is small. So obviously the beam of light from the laser should also be thin. Okay. So, and it can, as I said, it's easily focused on a small area like a sample stream. And for these reasons, we prefer the lasers, right? And initially we used gas lasers, but now we all use solid state lasers. Almost everyone is using a solid state laser. And these are the different laser lines. So our different lasers, what we use. Okay, when I say laser line, this is the ultraviolet laser, which emits ultraviolet rays at a wavelength of 355 nanometer. Then you have violet laser, which emits light between 405 nanometer. Then you have the blue laser, you have the green laser, yellow laser, and the red laser, right? The most common laser used in flow cytometer is blue laser. So if you take older machines like a 4-color instrument or a 4-color instrument, they use blue laser. The next commoner one is the red laser. So if you take a 6-color instrument or an 8-color instrument, they use blue and red, right? Then if you go for a 13-color, the third laser which is commonly used is a violet laser. Now you got more 20-color instruments, 25-color instrument. I'll come to color. What is it? I'll deal it later. But you can use yellow, green, and ultraviolet also. Now many things have come. Okay, the most common one is blue, red, and violet. Okay, and every laser can excite its corresponding fluorochromes. So fluorochromes are substances, are tracer elements. Because it's a 1 hour lecture, I cannot go very deep into fluorochromes. Fluorochrome is something which gets excited by that corresponding laser line, right? For example, you cannot excite a fluorochrome called FITC with the ultraviolet laser. Similarly, you cannot excite a fluorochrome called Pacific blue with a blue laser. So the laser line and the fluorochrome should match, right? So the laser arrangement in the machine is also different. Okay, there is something called collinear arrangement, which is a beautiful where the blue, for example, here I'm showing two laser lines, the blue laser line and the red laser line. Collinear laser arrangement is where the laser light, both red and blue, hits the cell at the same point of time, because it is very close and it is hitting the cell at the same point of time. It's a collinear laser arrangement, which is great. Whereas the other laser arrangement is parallel laser arrangement, where you can see that there is a blue laser and below that there is a red laser, and this is the cells which is flowing. So every cell will get interrogated first by the blue laser, and after a period of time it descends down and gets interrogated by the red laser. The difference between these two time points is called laser delay or time delay. Now you tell me which is the best configuration, collinear or parallel? Technically, which I would love to pick. I would like to pick collinear laser arrangement, not parallel, because see, I want the data from that cell at that point of time. Here, this particular neutrophil, for example, gets first hit by the blue laser. So all the fluorochromes which are excited by the blue laser will emit signal, and then it has to come down, and then it will be interrogated by the red laser, where now the fluorochromes which are excited by red lasers will give signal. So there is a time delay. You will not collecting signal at the same point of time. Blue signals first, I mean fluorochromes excited by the blue laser give signal first, and then fluorochromes excited by the red laser give signal next after a time gap, which is not so great, right? So, but many people use parallel laser arrangement, and they do something called, and this is again set by default, like mostly the user cannot change this much, only the service guy can have an access to it. And this loss, or they make a correction in a parallel laser arrangement in such a way that both they reduce this time delay and make sure that even in a parallel laser arrangement, the signals from the fluorochromes of blue and red are captured at the same time. They make a correction. That correction is called laser delay. Okay, this laser delay represents the amount of time it takes a cell to move from one laser to next laser in the relay. Okay, and it will be corrected, and the laser delay should be corrected in such a way that the cell interacted with all the lasers at the same time. Okay, so in collinear laser arrangement, you need not worry about laser delay setting, but in a parallel laser arrangement, you need to fix this. And again, I'm telling you, this is not under the control of the user. It will be set default by the manufacturer, and only the engineer has access to it, right? But you can monitor with your QC beats. You need to monitor daily with your QC beats. If things are not good, then you need to call your service engineer, but you can't do your own tweakings here, right? So this is about laser delay. Say when you're not setting your laser delay perfectly, what happens? So this is, you can see this cell first interrogated by the red laser, you get a signal, and then blue laser, you get a signal for the blue fluorochrome of the blue laser, and then you get a signal for the fluorochrome of violet laser. Now, if you're not correcting the laser delay correctly, you see signals at different time points, right? Which you don't want. So this is a good example. See, this is a data where the laser delay is not corrected. Okay, you can see loss of signal. You can see actually this is a 100% double positive case, where out of which 11% signal is lost because the laser delay is not corrected. Whereas when you correct it perfectly, then you will get a perfect data. Okay, so laser delay, you should know what it is. You should able to monitor it during running your quality control beats. And when there is a problem, you should call your manufacturer. You cannot do this tweaking, or you can't say like, I'll correct the laser delay. No, it's beyond your capability. Right? So that is about the light source. So just to summarize light source, the common light source used is solid state laser, and you have multiple laser lines, like blue is the commonest one, red, violet, then you have yellow, green, then you have ultraviolet also. And the number of lasers which is present in your machine depends upon the number of colors your machine can detect. If I'm using a 13 color flow cytometer, my machine instrument have 3 lasers, blue, red, and violet, and I should have 13 detectors to capture signal from 13 different fluorochromes, right? So that is about the laser light source, and you should know what is laser delay, right? So any doubts till now? Can I go for the next component? So we are finished with flow chamber, fluidics, and light source. Now we need to go for optics. So any doubts till now? Is there any questions on the chat box, or any participant want to unmute
No
and you can ask your
questions
question?
from the chat box, but yes, we do encourage if there's any kind of question up till now, please raise your hand.
Yeah.
You could even, just punch onto the React button where you can just
Yes.
raise your hand, and then we could just pick you out and take your questions.
Right. So if you have
Sir,
questions, you stop me. Yeah.
is there any thing like this? The sample has to have this much of total counts. In case of cytopenias, when the total count is
Okay.
very low, can we get that sample for flow cytometry, or is
Yes,
there
you
any
can. You can.
limit?
Now, I'll tell you one instance where you cannot take the sample when the count is very low. Normally, okay, now let's going into the practical aspect. Will be, I suppose they are organizing another session where it's more of practical flow cytometry. But let me tell you, even if you have, I'll tell you what, with we have achieved. There is nothing like you should reject when the sample is hypercellular, especially when you're dealing with peripheral blood and bone marrow. There will be enough number of cells for you to process the sample. All you need to do is increase the volume of sample. Say regularly, if you're taking 100 microliter, 200 microliter for processing, or 500 microliter.
Okay,
Now,
sir.
if it's hypercellular, take more volume of sample, lyse it, you'll get the pellet. One area where we find it difficult is CSF samples, what we get in leukemia patients. What happens is when the count is very low, say for example, 0 cells per microliter or 10 cells microliter, then that makes it difficult. Even because you can't get more volume for
Yes.
CSF, right? They hardly give you some ml, 1 ml or 1.5 ml, 2 ml. So you may not able to get that cellularity. And when you don't have that good cellularity, it doesn't form a cluster. So
Yes.
only when it forms a cluster, you have your confidence of calling it because of LLOQ, other things. When it's just dispersed dots, then you don't know whether it's a debris or a real cell. So you can't do that. But bone marrow and peripheral blood, I feel, yes, you can do it. All you need is slice more amount of sample.
Yes.
That's it. Right. Let's go into optics now.
Yes.
This is going to be
Thanks.
very interesting. Now, I told
Yeah.
you that, so these are the cells. It is made to come in a single file pattern because of hydrodynamic focusing, because of a faster moving sheath fluid, which at the funneling area pushes the cells to the center and all falls one after the other in a single file pattern, right? And you have the beautiful laser, the solid state laser, which is high intensity, coherent, monochromatic, hits the cell. So when it hits the cell, what happens? Two things happen: scatter, right? You have forward scatter and side scatter parallelly happening. And you also have the fluorochromes, which are attached to the cell to glow or emit signal, right? So have you ever wondered that, see, if I'm using a 13 color flow cytometer, let's assume I'm using a 13 color flow cytometer, forward scatter, no problem, because the light hits the cell and the forward scatter is collected in the same direction of the light source. So this is the forward scatter detector, fine. But whereas coming to the fluorochromes, 13 fluorochromes are now sitting on the cell, right? 13 fluorochromes are attached to the cell, and all the 13 fluorochromes will emit signal, okay, of different colors. It will be like a disco, right? Like 13 colors are getting emitted. Have you ever wondered how can we filter this and collect the corresponding signal from every specific fluorochrome? For example, 13 fluorochromes, I can say fluorochrome A, B, C, D, E, F, G, H, I, J. So like, how do you collect signal from fluorochrome A alone? How do you collect signal from fluorochrome B alone? Because it's all a mixture here, right? So that is the reason why we use filters, right? Filters. Now let's see what are these filters is all about. Optics, which is nothing but filters. So the filters what we use in flow is called dichroic mirrors. Okay, now this dichroic mirrors has two properties. I repeat, di means two, right? So it has two properties, dichroic mirrors. First property is it acts as a beam splitter, which means it allows a certain amount of light to pass through and a certain amount to get reflected. I repeat, first property is by its act as a beam splitter name, no, the name itself tells it. So it allows some of the light to pass through it and some amount of light to get deflected or reflected, right? That is first property. The second property is even that allowing of light, you can set a range. For example, if it is a long pass dichroic mirror, so it allows light of specific wavelength, more than a specific wavelength, to pass through. So I'll tell you an example here. So I'll go here. So let's assume that this is a long pass 750 dichroic mirror. Okay. Now, how does it does? It allows light of wavelength greater than 750 to pass through it. Okay, long pass 750. So it allows light of wavelength greater than 750 to pass through it. And all the light of wavelength lesser than 750 will be deflected. Superb, na? And you similarly, you have other dichroic mirrors called short pass dichroic mirror. So now this is a short pass 500 dichroic mirror. So name says, right, it allows light of wavelength lesser than 500 to pass through it, and anything more than 500 will be deflected. Beauty. And then you have band pass dichroic mirrors, where this is a band pass dichroic mirror 525 plus or minus 50. So this allows light of wavelength between 475 to 575 to pass through it, and the rest all lower than 475 will be deflected, greater than 575 will be deflected. So this is band pass. A specific range will be allowed to pass through the band pass filter. So with this beautiful dichroic mirrors, you can able to filter the light, right? Because all the 13 fluorochromes, with the example what I'm saying. So for, let's take a simpler example. So you have a 4-color flow cytometer. All the four fluorochromes will emit signal, right? Now we need to filter fluorochrome A, fluorochrome B, fluorochrome C, and fluorochrome D, and you use dichroic mirrors for that. So see, this is again a beautiful image. So here is a light which is a mixture coming from all the fluorochromes A, B, C, D, so and so. And now if you place a short pass 575, all the light which is lesser than 575 will be allowed, and the remaining all will be deflected. Similarly, 520 long pass, it means any light of greater than 520 nanometer will be allowed to pass, and shorter light will be deflected. So everybody knows this VIBGYOR, VIBGYOR. Violet as the lowest wavelength, red as the highest wavelength. So you can see the red is allowed to pass, right? The violet is all deflected. Similarly, you see this one 530-20, which means it allows light of wavelength between 610 to 650 to pass through, which is red light, and all will be others will be deflected. Okay, so again, the dichroic mirror short pass allows light of wavelength lesser than 540 to pass through it, and all other longer wavelength light is deflected. Beauty, right? So this is a dichroic mirror, and again, I'm just gonna say the same, right? You know it now. So this is long pass 500, which allows light greater than 500 to pass through, and the rest all will be deflected. And short pass 500 allows light lower than 500 to pass through it, and rest all will be deflected. Band pass allows a specific range of wavelength of light. This is 480 to 520. So that allows only this blue light to go, and all other gets deflected. So how does it been arranged in the flow cytometer? That's how it's arranged. So now you have the laser, you have the cells falling in a single file pattern. The laser hits the cell, forward scatter gets recorded. That's not a problem. The side scatter and the fluorescent light will be now going like this. The side scatter and the fluorescence light. Now let's assume this is a 4-color flow cytometer using fluorochrome A, B, sorry, this is a 3-color. So fluorochrome A, B, and C. Okay, so all a mixture of 3 light is going here. First is goes and hits the filter. Now this is a dichroic mirror which allows short pass. Okay, so red light gets deflected, and all the other light is getting allowed to pass through it. This is a short pass filter. Again, this is another short pass filter. So green and blue light is allowed to pass through, whereas the yellow is getting deflected, and it will be captured by the corresponding detector. And then you have this final detector which will capture. Now you see from this mixture, you are beautifully filtering the red light, the yellow light, and the green light. So that is the use of filters, basically to filter out the fluorescent light. Okay, and this is actually a layout of a very old flow cytometer, 4-color flow cytometer, which is called EPIX XL, Beckman Coulter EPIX XL. And you see here, this is the laser. Okay, and this is the flow cell or the flow chamber, and the light hits the cell here, and forward scatter is collected by the detector, forward scatter detector, whereas the side scatter and the fluorescent light, it goes here, and now you have the filters. So this is a long pass filter which allows long pass light, and shorter wavelength, short light is captured by the side scatter detector. Similarly, the filtering happens. These are all long pass filters, right? The graduations increases slowly. Say for example, this is 500 long pass, this is 550 long pass, 600 long pass, 650, and finally the red light, which has the longest wavelength, is captured by that corresponding detector. So this is how the light from different fluorochromes is filtered and captured, right? This is again the spatial arrangement. You see, this is beautiful. You see, this is the light. So, and you need to know that before a detector, normally there will be a band pass filter. So you have a long pass filter here, and after that you also have a band pass filter, which will typically collect a pure light of that wavelength, right? So all the deflectors, so you see there, this one comes here, goes through all the deflected light, then goes here, then some pass through it, and the other gets deflected, that comes here. So that's why you beautifully separate out the signals or the color light from that specific fluorochrome to that corresponding detector, right? Beauty, na, how they have designed. So excellent. So, and this is the outline, like I have shown you a pictogram, and this is the actual how it looks in the machine, right? Actually how it looks in the machine. So you have the different, this is band pass, this is long pass, LP means long pass, BP means band pass, or if you say plus or minus 26, this is band pass, and you also have short pass filters if needed, right? So that is about the filter. So this is what happens exactly. So the cells are passing, the laser hits the cell, the forward scatter, no problem, whereas the side scatter and the fluorescent light just goes, and the side scatter is captured by the side scatter detector, and the fluorescent light is filtered one after the other using your band pass, short pass, and long pass filters, and it is taken to that corresponding detector. See, this is 1, 2, 3, 4, 5, 6, 7, 8, 10, 8 color flow cytometer, right? This is 4 color flow cytometer. So that's about the filters, right? So then now we go into the detectors. Any doubts with optics? Is that clear? Is that clear?
Yes.
Thank you. Superb. So now let's go for the final part, that is the detectors. So we have studied about the flow cell and the flow chamber. We studied about the laser. We studied about the filters, which filters this light and takes it to the corresponding detector, right? Now let's study what are these detectors. So normally detectors are of two types. One is a photodiode, okay, and the other is photomultiplier tube. Now, can anyone guess where do we use photodiodes and where do we use photomultiplier tube? And you need to tell me why. So you have lights from forward scatter, you have light from light for side scatter, light from side scatter, and you have light from fluorescence, or the light from the fluorochromes emitted. Now, where do you want to use photodiodes? Where do you want to use photomultiplier tube, or you can use randomly any detector? See, okay, let me put this way. Among all these lights, the light, the forward scatter light, the side scatter light, and the fluorescent light from the fluorochromes, among this, which is strong? Which is relatively strong? Yes, Hari? Yes, forward scatter. Why? Because forward scatter light is in the same direction of your laser light source. Obviously, it will be strong, whereas the other light is all a part of scatter. For example, forward scatter will be almost like predominant of your light, incident light, the from the laser, right? Whereas the side scatter and fluorescent light will be almost like 1% of it, right? I'm just telling you 1%, okay? So it is very weak. So forward scatter light is very strong. For such a strong signal or light, you should use a stable detector, strong detector, which can able to withstand it. Such a detector is called photodiode. Photodiode, which is strong, okay, stable, and it can, for heavy signals, this is great. High intensity signals, photodiode is good. Whereas the side scatter signal and the fluorescent signal are very low intensity, weak, right? In such a case, you want a more sensitive detector. It doesn't matter about stableness now. You need a more sensitive detector. So photomultiplier tube is a sensitive detector because it can multiply the photons, whereas photodiode normally doesn't multiply. Okay, so it can amplify the signal. So you need, for a weaker signal like side scatter and fluorescent signal, you need a very sensitive detector which can multiply photons, which is called a photomultiplier tube. Is that clear now? So forward scatter signal, you will be normally using a photodiode, and for side scatter and fluorescent signal which are weak, you use photomultiplier tube. Now photomultiplier tube is sensitive, but it is not stable. Now if you open the instrument without the service engineering saying like I'll repair today, when the machine goes wrong, you will damage the photomultiplier tube as it gets exposed to the daylight or any light, because it's very sensitive. And with the current on, when the machine on, when you open the machine, if the photomultiplier tube is exposed to the light, it's gone, finish. Now instead of repairing, you're going to make a big expense, right? So photomultiplier tube is very sensitive, so you should not expose it to light, right? So now suddenly why I'm showing this, what is this?
Solar
I want to
panel.
take you,
Solar panels.
sorry.
Solar
panels.
Very good. So when I'm talking about flow, to make you get back to the flow, I'm just taking this odd out. Is it really odd to the topic now, or you can connect it? Why I'm showing a solar panel now? What is a solar panel?
It is also a detector.
Very good. What type of detector is that? It's a photodiode.
Yeah.
This is a photodiode. Okay. What does a solar panel does? It converts photons to electron. What your detector does? It also converts photons to electrons. Simple. Now you see this is a photodiode. It is stable. That's why you kept it on the terrace. Okay. So it's a very stable detector. So sunlight falls on it, right? And it converts the photons to electrons. So this is a photodiode. It's okay. So that's what the common thing. So see, forward scatter signal is very high intensity signal because in the same direction of the light source, right? And you can use photodiode for it, right? And if I want to discuss all these things, you go very crazy. I'm not going to go into that physics. Okay. And this is a photodiode, how it there in the machine. So this is photodiode, which is very sensitive, used for capturing high intensity light like your forward scatter light. Whereas photomultiplier tube is used to capture weak signals like your side scatter and fluorescent signals, because they have the inherent capacity. When you give electric current to the photomultiplier tube, it can amplify the photons. Okay, it can amplify the photons, and it can generate signals. So even if one photon impinges, it can multiply and make a considerable signal. Whereas if just one photon hits the photodiode, it doesn't even respect that. Okay, so that is the reason for weaker signals you photomultiplier tube, and for stronger signals you use photodiode detectors, right? But the problem with photomultiplier tube, I'll tell you, it is working with the current, right? Even if there is no signal, sometimes even if there is no actual photons are eating it, sometimes because of some electrical thing, it can give you some weak signal, which we call it as dark current, which is noise. Basically there is no signal, but still because of the multiplication, it may give you signal which is called dark current. Okay, now what is the problem with this is, because it amplifies so heavily, the problem is, see, this is a case where it's a fluorescent signal, right? Which is obtained from a photomultiplier tube. Now, if I increase the voltage of this photomultiplier tube, even this negative signal, because of amplification, okay, it can mimic like positivity. You can push that signal to positive region. And this is very important. Once it happened, like before, very early when I was using flow before some 12 years, my technician, what he did, I was eagerly waiting for a signal for one particular marker, which is tagged to a fluorochrome APC. So he saw that it's coming negative. I'm asking him to repeat. So what he did, he slowly adjusted the voltage of that particular APC channel, that is APC detector. So now because of this high voltage input, now the amplification is more vigorous. So though there is no signal, which is noise, now the noise is amplified because of dark current, and it can mimic like a positivity, but actually it's not, right? So you should be very careful when you're working with flow. If you don't know the basics, Normally what we do is, I'll tell you, I don't know how many of them fall in that category. Even I was in that category some time back. Like, we normally go into analysis first. We start analyzing cases, leukemias, lymphomas, and so and so. We never learn the basics. We don't never go in a step-by-step basis. First, you learn the basics, compensation, all those things. We directly go into analysis, and retrospectively we learn all this, right? But it's very good that you learn your basics and then go for analysis. That's the best way to go about it, right? So otherwise you can make, see this, if you're not knowing the basics, this you can call it as false positive, and you're gone. You're going to find out a bad report, right? So that is a thing with photomultiplier tube. You should have a check on the voltage of a photomultiplier tube. And now recently, especially Beckman guys have come with another detector, which is stable as a photodiode, but can also multiply the photons. So it is a combination of both conventional photodiode and a photomultiplier tube, something like, and that's called avalanche photodiodes, APDs. The machine which they came up with, DXFlex, has this particular detectors to them. So here also the same problem. It's going to amplify the photons. There in PMT you use voltage, here you use gain. So if you increase the gain, then you may have the same problem what you had with PMTs, but it is more stable than PMTs. That is something which you need to know. But you need to remember that this is APDs, which is used in the recent flow cytometers of Beckman-Coulter, right? So now coming to this point, what does this color mean? Normally people, when we discuss, they say, no, I'm using a 13 color flow cytometer. Some people say it with great pride, 13 color flow cytometer. I use a 20 color flow cytometer. Some people say I'm using 8 color. What does this color mean? Anyone? So the color depends upon two things. What are those? One is the laser. For example, if you're using one laser, how many maximum number of fluorochromes you can excite with one laser? Maximum 5 with the current fluorochromes available, right? So, but if your instrument has only one laser, say blue laser, the maximum number of fluorochromes which you can excite is 5. So it's a 5 color instrument. And remember, when it's exciting 5 fluorochromes, you should have one detector corresponding to every fluorochrome. So you should have 5 detectors. Now, if someone says I'm using a 8 color flow cytometer, now the maximum number of lasers you need is a 2 lasers, the blue and red, and you should have 8 detectors to capture signal from 8 different fluorochromes, right? And someone says 13 color flow cytometer, you should have 3 lasers, blue, red, and violet, and you should have 13 detectors for 13 different fluorochromes which you can add to the cells, right? So that means a color, the maximum number of colors which your machine can capture or which you can use it in your experiment is called the colors. Okay, so you have 4 color instruments, 5 color instruments, 6 color, 8 color, 13 color. Now it has gone up to 40 color, right? There is spectral flow, which is a different topic, but this is what color means. So which machine you want to use? 8 color, 10 color, 13 color. It depends upon the experiment. It depends upon the experiment. For example, now if you are using, if you want to do a simple lymphocyte subset analysis, 4 color instrument is fine. But if you want to do a complex experiment like your leukemia, lymphoma, cytokine profiling or something, you need a higher version of instrument like 8 color or 10 color instrument. Okay, so normally people get happy, more colors, more merrier. Yeah, but everything comes with a price. What is the problem when you go for high color instruments? Yes, Hari. Hari is an expert.
Sir, compensation.
Yes, compensation. Everything comes for a price. You know, when you go for multiple lasers, then you need to be a king of compensation setting, because there will be a lot of mixing. You should do a good compensation workup. You also need to do post-acquisition compensation, right? You should be compensation may be challenging as color increases. So never be too excited that I'm using 40 color. You should be very good in doing the compensation. In spectral flow, it's a different story. Their unmixing can be done automatically, but here you need to burn conventional flows. You need to do your own compensation. So now we finished about flow chamber, fluidics, light source, optics, detectors. Now finally we're coming to electronics, which is more of physics. I'll make it simple for you. Maybe if in 5 minutes I'll finish. I'm sorry, people who are holding on. So the detector, basically what it does, I told you, the photons will be converted to electrons, right? The electrons will be filtered, and all the analog values will be converted to digital values. So analog to digital conversion, and there is something called digital signal processing. Okay, so the digital signals will be processed, and finally data by numbers will be generated. And as I said, everything will be shown on a dot plot with a dot, which is signal from every cell, right? So you see, this is, I'm just showing you, this is pulse processing forward scatter. See, cell goes, you get the signal like this, right? So for every parameter, you get a signal. Forward scatter, for every cell, you get a pulse. Side scatter, every cell gives a pulse. Fluorochrome A gives you a pulse for one cell. So if you're analyzing 1 lakh cells, for forward scatter, you get 1 lakh forward scatter pulse. Similarly, 1 lakh side scatter pulse. Similarly, 1 lakh fluorochrome 1A pulse, fluorochrome B pulse. So you get so much of data, right? So that is called the data, right? And you have to analyze this data with a software. Okay, it can be a third-party software and acquisition software. See, this is a, so now assume that this is a cell which is in line with the laser, and these are the fluorochromes. So when you have lesser fluorochromes attached to the cell, you get a weaker pulse. Height of the pulse will be short. When you have multiple fluorochromes attached to the cell, the height of the pulse will be more. When you have bright, lot of fluorochromes attached, the height will be much more. So height will always tell you about the brightness of the signal. Remember, brightness of the signal, and the width of the pulse will be denoted by the size of the cell. For example, if it's a large cell, the time period it's an interrogation with the laser beam will be more, so you will get a wider pulse. Okay, so height, fluorescent intensity, and width is size of the cell. So every parameter, every pulse will have three things: height, which tells you about the interfluorescent intensity or the signal strength. You have the width, which tells about the size of the cell. And when you combine this, you get area. So even if it's a forward scatter pulse, you will get three values: area, width, and height. Similarly, for side scatter, all these three fluorescent parameters, everything three. And remember, for every cell, these three is collected for forward scatter. Similarly, for every cell, side scatter, these three parameters are. So the data is huge, right? So when you acquire large number of cells, 10 million cells, the file size used to be so big that because all these numbers are calculated, are captured, right? So that's what happens basically. So laser beam hits and the detector captures it, and all this analog to digital conversion, baseline processing, everything, pulse generation, pulse processing, everything happens. And then what you see in the computer is analysis. Very simple. That's the most sweetest part people see, but these all people don't look at. Okay, but if anything goes wrong in this area, this is absolute crap, absolutely useless to do it. Okay, so you should know all these things before you go to this analysis part. So this is the analysis. So you have softwares to analyze. So this is a dot plot. As I said, every dot is a cell. Like dots make a cluster, and they call it as population. So this is a dot plot where you can see 4 populations. This is positive for CD3, which is tagged to fluorochrome APC, the x-axis. This is positive for fluorochrome P, which is tagged to the marker CD56. This is double negative, and this is double positive. Similarly, you can show the data by histogram, where you have a positive population and a negative population. And this is a dim population. The beauty of flow is, in contrast to IHC, in IHC, what do? You say binary, positive, negative, that's it. In flow, you can able to say negative, positive, and positive, you can say grade it by intensity, say dim positive, moderate positive, bright positive, something like that, because you have that facility here, right? And all this positivity is because based on the fluorochromes, which I just showed you. If only cell has few target antigens, it will be dim. When has more target antigens, it will be bright. It's all based on the number of fluorochromes which attaches to the cell, right? So here again, so this is 2 fluorochromes are stained, it goes and attaches to the cell, and you get the data like this. This is positive for both, this is negative for both. This is positive for the y-axis marker, here it's APC, which is a fluorochrome allophycocyanin, and here it is positive for FITC fluorochrome, which is fluorescent isothiocyanate, right? So that's how beautifully happens. See, this is animation just shows you, this is a cell which is stained only with green fluorochrome. So you have here, so green falls here. When it is stained with only orange, red color, it falls here. Okay, so similarly, data is processed like that. This is multicolor flow cytometer. What I'm showing you is just 2 colors, but similarly, you can use 13 color, 20 color based on your configuration of the instrument. So coming back and summing up, what exactly happens in the flow cytometer? Cells has to go in a single file pattern. Very important. So here after single file, not lobular carcinoma. It is hydrodynamic focusing of flow cytometer. Focus light will hit the cell where scatter of light will happen, forward scatter, side scatter, and also parallelly emission of fluorescence from the fluorochromes attached to the cell will happen. And this light should be filtered and collected, right? Which is done by optics. Third, conversion of photons to electrons, which is done by the detectors. And finally, the data should be analyzed with a specialized software. So this is done by fluidics, this is done by laser, light source, optics, and finally electronics, and then interpretation with your software. Beauty, that's the flow, right? So beautiful. Now just, can I just finish in 3 minutes or 4 minutes? Can I have
Sure,
that?
please go ahead.
Yeah, thank you. So just few advancements. Now what I showed you is all conventional flow. What are the advancements which has come in? So next level multicolor flow cytometer. So normally now we go up to 10 color, 13 color, but there are multiple other instruments now like spectral flow cytometers and multicolor flow cytometers. Even multicolor conventional flow cytometers are out in the market. Flow sorters where you can sort out the population of interest. For example, from the blood, I want to work only with CD4 positive T helper cells. Now I don't want any other cells. I want only the CD4 positive T helper cells. With the flow sorter, I can able to sort out that helper cells and I can carry on my experiment with that special cells alone. So that way you can sort your desired cells from the sample. Then you have beautiful imaging flow cytometers where apart from the scatter and the capturing of fluorescence, you can also capture the image of the cells, right? Why you need microscopy? See, now morphology, right? Here also you can get the morphology, but not as great as your microscopy, but still you can get the images. Then flow fish, and then flow for nanoparticles and extracellular vesicles. Even now this is coming up in a great way where because patients with positive extracellular vesicles, right? For example, if you have a breast cancer patient and in a peripheral blood, you could able to find extracellular vesicles which are EpCAM positive or which is pertaining to cancer, then you can say that this patient prognosis is going to be bad because already there is extracellular vesicles circulating in the blood. And this patient may have a metastasis or may future have a metastasis likely. Now this is a 9 laser 50 parameter instrument. So it has 9 lasers. Remember, all I spoke about is blue, red, violet, yellow, green, and ultraviolet. There is 3 more laser lines and 48 fluorochrome marker, 48 detectors to capture signals from 48 fluorochromes and forward scatter and side scatter. That makes a 9 laser 50 parameter instrument. This is Symphony from BD. And cell sorting, I told you, right? If I want to collect only one particular population of a mixture of cells, now I can use a sorter. For example, now let's assume this is the population which I want to sort out. I don't want this neutrophil, lymphocyte, the monocytes, basophils. I just want CD4 positive T cells, which when I just get that and say sort out, it can give me that pure CD4 positive T helper cells for which I can use it for further experiments without contamination from other leukocytes, right? That is about sorting. And then see, this is how beautifully it sorts with the electric charge plates. So sorters are quite well available both from Beckman Coulter and BD. And imaging flow cytometer. So now what this imaging flow cytometer does it, I told you detectors, right? But apart from detectors, in imaging flow cytometers, you have cameras where it can capture the cells. So that gives you image of the cells. This is a beautiful image where you can see what they have done. This is a beautiful paper of flow fish. Let me quickly go through this. So you see, this is the image of the lymphocyte, bright field image. And what they have done is this is flow fish. So they've used a chromosome enumeration probe 17. Okay. And they've also used a 17p, which is especially for tagging a TP53, right? Now, if you see this cell, you can see that there is 2 signals of chromosome 17, which is normal. So here they're using probes which are used in fish, fluorescent in situ hybridization, and also they are using fluorochromes, which I was talking about. So you can see that this is normal. So it has 2 chromosome 17 and 2 intact TP53. And you can see this is a viable cell because cyto, it is CD3 positive. So basically it's a T cell, 5 is also positive, right? So again, this is something. So if say, for example, now if a cell has there is one signal lost here, for example, now this signal is lost. Now you can able to say that apart from saying this is a T cell in a neoplastic T cell, you can also say this cell as a mutated TP53 as seen here. So this is a case of CLL. You can beautifully see that there are two chromosomes 17, which is normal, but only one TP53 gene is there. The other TP53 region has got deleted. And you can see that this is 19 positive, so it's a B cell. It is 3 negative, 5 positive. So if you have 19 and 5, it should be a mantle cell or a CLL, right? This is a CLL case. And you can say, just saying CLL, currently we say just CLL. But when you use imaging flow cytometer, which has this, you can say it's a CLL with a deletion of TP53, which carries a very worse prognosis. That's the beauty of imaging flow cytometer. You can see this one copy of 17p region corresponding to the TP53 region is gone, whereas the chromosome is intact, right? So that's a beauty. So that's imaging flow cytometers. Applications, I'll just finish off. So now there is a lot of applications. Primary immunodeficiency disorder is actually a group where you have a lot of flow assays there, starting from a basic lymphocyte subset, extended lymphocyte subset, DHR, LAD, DNTs, VAS, HLH, so and so. Look, but whenever we ask applications about flow, people say leukemia lymphoma. Yeah, leukemia lymphoma is there. That is almost there. But beyond that, there is more other applications like PID. PID is coming up in a great way. Transplant immunology, stem cell enumeration, fetal-maternal hemorrhage, PNH, platelet function disorders, immunotherapy, and very importantly, currently cell therapy. Cell therapy is coming up in a big way, and flow has a lot of potential in that. Right? MRD is one thing, which is again very important. One of the most taxing assays of flow is MRD, where you need to sit patiently and detect that 0.001% residual disease. Right? To conclude, so it's a powerful tool that enables rapid. One beauty of flow is rapidity. You give the sample in the morning, I can give you the report in the evening. Right? Whereas the other assays like NGS and molecular, I don't think so it's possible. Multi-parametric analysis of individual cells. And one more beauty of flow is it's a cell analysis. You are analyzing an intact cell. You are not taking anything out of the cell. Molecular assays, there is always a problem with contamination because you remove the DNA off. The cell is no more with the DNA. All the DNA you pull and do, and some contamination of DNA gone. But flow, it's a cell-based analysis. It's like you analyze the cell, right? And it provides valuable insights in immunology, molecular biology, and cancer research and infectious disease monitoring, right? And as I said, it helps in identification and characterization of diverse cell populations, because here you are exploring both physical properties as well as the antigens and proteins present in the cell. And enhanced cell phenotyping, more precise immune profiling by flow. If you have say, for example, the example what I showed you, a 40-color flow cytometer, you can clearly dissect out your immune system and study them to the precise of immune profiling and the cytokines what it can produce. And finally, it remains an indispensable tool, continually advancing technology that will undoubtedly continue to expand its impact on discovery, clinical applications, and our understanding on cellular complexity. So flow is going to go a long way, I'll tell you. It's going to be a lot of new updates are coming up, like spectral flows are coming, and it's going to make a roaring advancements in the future. With that, I thank you for the opportunity. It was great. I don't know how about your side. I enjoyed it talking to you all.
Thank you so much, Dr. Thulasi, for that amazing, spectacular, and invigorating talk on flow cytometry. Before I would like to say anything further, I would encourage to see if we could have any kind of questions or feedbacks or suggestions on the topic. We'd be happy to take any questions. Dr. Thulasi, I'm sure would be very happy to take those questions.
Yeah, sure, 100%.
Any questions, anything is welcome.
Anything, we can say anything related to flow cytometry. I'll give you that liberty.
Yeah, we're getting a lot of comments on the charts about thank you for an excellent session, very informative and helpful. Okay. For informative, great presentation. Thank you so much. Nice presentation. While all of that is great, if you all have some particular specific questions, Dr. Thulasi will be more than happy to take them.
You can ask anything. Don't any basic doubts. Yeah. No problem. If you have any other doubts, you can connect me with this IDs. Okay, I'll be there. I'll be happy to take up your questions or anything, any flow related doubts. I'd be happy to retake. And again, I want to thank Dr. Renu for giving this opportunity. It's like
we would like to thank you, Dr. Thulasi, for I've dealt with flow while I was working in Healthcare Global Cancer
I
Hospital
know.
myself. But today's session was incredibly refreshing, and even for me. And as you correctly pointed out, it's not just important that we know how to interpret the flow scatter plots, right, in cases of leukemias and lymphomas, and that we do that, right? And it is so important to know about the instrumentation, the fundamentals of the mechanisms, in case we get stuck, and especially during root cause analysis and trying to troubleshoot when you have a very tricky case. And flow can be very tricky many times. There are a lot of gray zones as well. And that's where a thorough knowledge of the instrumentation, the principles, the mechanisms, and all of that also matter, and not just about the interpretation of those scatter plots.
100%.
And that's precisely why we had this topic today, to see what's behind the scatter plots, right? What's behind the dots, and that's so important. And as fluid as the sheath fluid in the flow cytometer, your talk was as fluid as that today. I thoroughly enjoyed it, and I'm sure each and every one of us who witnessed this talk of yours today thoroughly not just enjoyed it, but I think we're going to go back home more enlightened than we were before this session. And we look forward to a lot of talks from you, Dr. Thulasi, in the future. So for all of you present here in the talks today, we're going to have a lot of other practical talks, not just on flow cytometry, but on hematopathology as well, from our fantastic speaker today, Dr. Thulasi. And it'd be very nice to also know in the chat box if you have something particular that you would like to. Okay, we have a question here.
Yes.
So plasma cytomas can be detected with flow, yes or no, sir?
Plasma cytomas. Okay, now, so plasma cytomas, as you know, you have a clonal population of plasma cells in a particular location, right? So now the question comes is whether is that plasma cytoma, because you want to label as plasma cytoma only after ruling out myeloma. Okay, in that context, they do a marrow and send you. And if you find greater than 10% of plasma cells in the marrow, clonal plasma cells greater than 10% and other myeloma defining events, it is no more a plasma cytoma, it becomes a multiple myeloma. First question. Second, if it is not greater than 10%, you can make sure how minimal involvement it is there in the marrow. For example, I have seen plasma cytomas and plasma cells in the bone marrow by morphology maybe around 2 to 3%. Now what you need to do is you need to do flow and check whether that percentage of plasma cells is clonal or not. You can say that clonal abnormal plasma cell burden is so and so percentage. The clinician may keep a check of that, but currently I'll tell you in standard of practice, people are not doing flow for plasma cytoma. What they do the marrow, and when you say that the plasma cell is just 2 to 3%, they happily say, okay, then it's a case of plasma cytoma, and then go on. But what I feel is you need to check those 2 to 3% plasma cells by flow, check the clonality. If it is abnormal plasma cells, maybe it may be like 1% or 0.5%. You need to document that, because and you may also, after your treatment, you can check that again. If it is increasing, maybe the patient at some point of time may come out as myeloma, and he may need a systemic therapy, not just radiotherapy or localized treatment. So the indication currently in the standard of care practices, people are just doing morphology, they're not doing flow. But I would recommend them doing flow, because multiple myeloma, there's no point of doing flow in diagnostic time point. Okay, because you have a lot of plasma cells, you have the M protein, immunotyping gives you, and the clinically you have light occlusions done. Whereas initially people are not using flow for follow-ups also. Now IMWG criteria has come clearly stating that you need to do MRD for patients of myeloma on specific time points to take it for transplant or to stop the treatment and so and so. So plasmacytoma currently flow as a very limited role. People are not asking for. They are just looking at the bone marrow morphology, and if it is say 2% or less than 5% or something, they are happy. But if you do, you will explore a lot of things. If you do flow and you find a greater burden of clonal abnormal plasma cells of myeloma phenotype, then these patients may have a relatively poorer prognosis compared to patients who don't have any abnormal clonal plasma cells and just a plasmacytoma. I suppose I answered that question.
Yes, so she's mentioned that. Thank you for the clarification. We have another question from Dr. Shalini. In degenerated samples, is blast detected in flow better than peripheral smear or bone marrow examination?
Okay, so in a degenerated sample.
Yeah, are blasts detected, and probably that what she's trying to say is that are blasts detected through flow cytometry? Is that better on peripheral smear, peripheral blood probably, or bone marrow examination?
Okay, so if I understand the question correctly, you mean to take you got a degenerated sample and you make a smear out of it, the degenerated sample, and you want to run flow. Both will be bad, to be honest, because flow you can run up to 72 hours if it's an apparent sample, and beyond 72 hours you can run and try, but it may not give you good results. Better detected flow or morphology, I would say more of what to say, depends upon the percentage also, but morphology. Why degenerated sample? It's like you can try. You can try, because beyond you can try. If it's a good number of blast, you can able to come up with a diagnosis. If it is just 5%, 6%, or less than 10%, there may be a chance you may not give a confident report in that case. Morphology also, you in a degenerated sample, how confident you will be calling a blast. So I don't know that. It's tough. It's tough. But if it's good enough, it all depends upon the percentage of abnormal cells in a degenerated sample. If it's full of blast, yes, you can still go and give the report. If it is very few, then the job becomes difficult. You need a repeat sample.
Oh, thank you for that answer, Dr. Thulasi. We have Dr. Isha saying, can we have more sessions with clinical correlation and interpretation? It will be very helpful. We also have Teresa saying, can we have a presentation on compensation and troubleshooting and flow? Point noted.
Okay.
And yeah, these were some of the requests, I guess, that we have got from some of them in the audience. Can we have a session for compensation? I think compensation is something which is, again, a very tough thing to understand and quite challenging in practice. Probably we could take that up in the practical sessions that follow. That's also noted out over here. Any more questions from anyone else?
Can I make a comment?
Please go ahead, please.
Yeah, my name is Teresa from Ampath, Kenya. I think we are with the doctor in a WhatsApp group, and so he put the link there, and we joined. And so, first of all, I'm also very grateful that you've taken us to the basics. As you clearly said, many a times we go into interpretation of clinical data, and yet we do not understand how the instrument operates.
True.
So that is very important, and we I think we can collaborate more in future, so that we could, if your time allows, you could also give our students a talk on this basics of the instrument. Thank you very much.
Sure. And you're doing a great job in that group. I really appreciate that.
Thank you.
Any more questions? So I guess gradually we're coming to the end of the session. And I think all of us are more enlightened than we were before starting this session. Thank you so much again, Dr. Thulasi, Raman, for taking your time out on a Saturday evening and also during the festive season that is going around. And thank you so much for all the participants. A heartful, gratitude from all of us out over here for being present here and making this a success. We look forward to more engagement from all of you, and we will keep you posted on our LinkedIn platform. So keep watching that space out for the educative series from our side. And I wish you all a great evening ahead.